Hello, everyone, and a warm welcome to all of you who have joined us today. I am Bruna Lousada, the European Manager at Hansen, the Teledyne Labs division specialized in dissolution and diffusion test systems. And I'm thrilled to introduce today's session, Advances in Vitro Bioequivalence Assessment for Topical Products with IVPT focus, with Dr. Theo Capanazzi, co-founder and chief scientific officer of Daitiba. So this is the second part of a two-part webinar. The last session was focused on IVRT, on in vitro release test, and today we're going to focus mostly on IVPT, in vitro permeation test. For those who didn't watch the last webinar and are not familiar with Theo, he is the co-founder and chief scientific officer of Daitiba and has played a key role in establishing the company as a leading CRO in dermal and transdermal research services. With his guidance, Daitiba has established a strong track record in characterizing topically applied products and possesses superior scientific expertise in both in vitro release and in vitro permeation tests, and the methodologies developed at Daitiba have contributed to numerous innovator and generic drug approvals. So before we begin, a few housekeeping notes. So all attendees are currently muted, but you can submit your questions at any time using the question function on your screen. We will address as many questions as possible at the end of the presentation, but if we run out of time, we will follow up with you via email. If you experience any issues with your audio or connection, please try logging off and logging back on. For those who can't watch the entire webinar or wish to revisit it later, a replay will be available on the Teledyne Labs YouTube channel in the coming days. So with that, I'm delighted to hand over to you, Theo.
Thank you, Bruno. Thank you for the kind introduction and the opportunity to present today. Hello to anyone joining us online. I'm delighted to be here today to dive into the specifics of regulatory approval process of generic topical products submitted to US FDA and the EMA in the European Union. As you hear from Bruna, I'm Theo Capanazzi from Daitiba Analytical Services CRO based in Toronto, Canada. We have more than 20 years of the experience working with topical products performance testing, utilizing in vitro methodologies. Today, I will explore the significance of the in vitro permeation, IVPT, in developing and approving generic topical products. As you know, pharmaceutical companies must navigate stringent regulation, high cost, and extensive time investment to bring their products to the market. Demonstrating the quality, efficacy, and safety of the products is critical before obtaining marketing authorization from regulatory agencies. Therefore, having clear understanding of the specific requirements and consideration of each regulatory bodies is critical. As we know, the primary objectives of IVPT is to quantify the rates and the extent drug permeates through the skin, providing critical data to the formulations, potentially efficacy and safety. Establishing characteristics permeation profiles of the drug products when apply a discriminatory IVPT. IVPT data can be used to support bioequivalence by comparing permeation profiles of generic products with the reference drugs to demonstrate therapeutic equivalence, which is valuable for regulatory submissions. IVPT could help optimize formulations. By understanding how the drug permeates through the skin, formulators can optimize the composition and concentration of the excipients and active ingredients to enhance products' performance. It can screen formulation. IVPT can help screening multiple formulations early in the development process to identify those with the most promising permeation characteristics, saving time and resources. It predicts in vivo performance. While IVPT cannot replace clinical trials, it can provide predictive insights into how products might behave in a clinical settings, especially when correlated with the in vivo data. By understanding these distinctions, the objectives, the companies can effectively utilize IVPT as a part of their topical product development strategies. Role IVPT methods as a surrogate alternatives. IVPT assess drug permeations through the skin layers, mimic absorptions and distributions into and through the skin. It employs ex vivo human skin membranes to predict product behavior upon application. In terms of bioequivalence, IVPT demonstrates comparable permeation rates and extent to achieve drug absorption through the skin. Thus, IVPT evaluates drug availability at the skin site of actions, characterizing a comparing therapeutic equivalence between test and reference products. We do have the regulatory guidance showing here. EMA draft guidance on topical products quality equivalences that recently, September, just a few weeks ago, 2024, has been updated. This guidance provides more details to how in vitro and vivo models may be suitable for clinical data and the purpose establishing therapeutic equivalency in the stepwise approach. We have two FDA guidance for IVRT and IVPT effective 6 October 2022. A recently revised USP Chapter 7024 includes IVPT, offering in vitro performance tests for semi-solid and liquid-based dosage forms. Both FDA and EMA provide standard for validating and using in vitro tests for bioequivalence in NDA submissions. Benefits of in vitro studies: this is a cost-effective, time-saving alternatives to the clinical studies, offering controlled and reproducible bioequivalence assessment, accelerate products approval, reduce research costs, and speed up availability of generic topical products. Regardless, regulatory agencies may harmonize IVRT and IVPT methodologies. There are some differences. The major differences in agency approaches: the FDA gives detailed product-specific guidance, while EMA adopts a broader perspective. We have classification systems showing on these slides. The FDA and EMA topical drugs classify into 4 classes based on the qualitative and quantitative compositions, microstructure arrangements of matter, and in vitro release. The topical generic products classification and their eligibility for biowaiver is detailedly described in the presented FDA references and in the newly revised EMA guidance on quality and equivalency of locally applied, locally acting cutaneous products. FDA generic topical drug products falling into the category class 1 and 3, having similar release profiles as RLD, will be eligible for biowaiver, waive clinical endpoint studies. Generic products in the class 2 and 4 will be required conduct in vivo BE studies as per agency requirement. EMA BE can be demonstrated by for simple formulation by documented Q1, Q2, and Q3, and IVRT equivalence. However, if IVRT is not able to show equivalence, therapeutic equivalence could be conducted by IVPT equivalence. Nevertheless, when addressing complex semi-solid equivalency regarding local availability should be also demonstrated. Also, both agencies share common point. The scopes completely different. The EMA recommends on fit-all approach with only one difference, reporting the simple versus complex formulation. On the other hand, FDA proposed specific guidance each product. The generic topical dermatological drug products are classified as a complex generics and be following availability PHGs. Or alternatively, approach could be requested at the pre-submission FDA meetings. About system now, the recent guidelines from FDA, EMA, OECD, and the USP General Chapter 7024 outlines recommended instrumentation for in vitro permeation tests, IVPT experiments. Currently available systems include this automated aliquot volume replacement system, manual French cell diffusion cells, and flows through brown cells type system. Any of systems can be used for regulatory submissions in IVPT studies. Each system has specific advantage and limitation, and that should be considered when selecting equipments for a particular topical product IVPT study. The choice of suitable systems frequently has been driven by nature of the testing products. As an example, could serve recently published article about the IVPT clascoterone topical creams. References shown on the slides. Yeah, clascoterone steroidal anti-androgen used topically for treat acne is unstable and in aqueous solution at room temperature quickly hydrolyzing. This instability poses significant changes in assessing the rates and extent clascoterone permeate from accurately. These results of the study reveal that utilizing flow through diffusion cells with fractional sampling minimize clascoterone hydrolysis, providing more accurate quantification than vertical diffusion cells with aliquot sampling. Vertical static diffusion cells showed considerable hydrolysis, while flows through systems significantly reduce degradation. Also, in previous experiments across the various sites, comparison full volume receptor replacement versus aliquot sampling and automated versus manual system highlighted technical challenges that must be also to be considered. The selection of the suitable diffusion systems is vital, but next crucial steps is choosing a qualifying a skin membranes. EMA typically requires IVPT to be conducted using surgically obtained human skin rather than cadavers. The FDA accepts both cadavers as surgically obtained skins. Notably, currently, none of the FDA and EMA accept ex vivo animal skins for B assessment. Key steps in drug delivery through the skin includes release from the formulation matrix, penetration from stratum corneum, and moving into the different skin layers. Factors that affect bioavailability are skin barrier integrity, physicochemical properties of the permeates, and characteristics, vesicles, components, excipients. IVPT membrane types, we have this. There is a full skin grafts, including dermis and entire epidermis and entire dermis. Preserves most skin characteristics. And second, we have split thickness skin grafts, includes part of dermis, categorized by thickness. Thin, it's between 150 to 300 micron, intermediate 300 to 500 micron, and the thick, it's 500 to 800 micron. Easier to harvest and cryopreserve. Further, we have epidermal membranes separated by heat or enzymatically. Advantage and limitation. It's full thickness skin lipophilic compounds may be retained in the dermis. Epidermal membranes fragile, unsuitable for some mass balance techniques. The main concerns using ex vivo models, lack of blood circulation, exfoliation, metabolism may not be replicated. Enhancing penetration and potentials. Choosing the right carrier vesicles is pivotal. Matrix vesicle and API interactions play a key role. Besides those listed skin grafts, we have innovative models too. For instance, this showing like genoskin ex vivo human tissue, real human tissue collected post-surgery and maintained it up to 7+ days, closest alternative to the in vivo clinical tested. Applications in immunology, toxicology, efficacy testing, and more. And we have also 3D bioprinted human skin equivalence created using sophisticated 3D bioprinted technology, highly reproducible models for studying skin diseases in vitro, useful for autoimmune skin diseases and skin cancer research. Okay, before initiating methods development, it is critical step is to select appropriate supplier for excited human skin as a membrane for IVPT study. These sources can be from surgical procedures or cadavers, featuring healthy, normal, and burial component skin from donors at least 18 years old. The study protocol should detail harvesting process, thickness, and storage conditions. Challenges and consideration: donor-to-donor variability and intra-donor variability. Important skin thickness consistency. Burial integrity test essential for membrane qualification. Avoid using compromised skin for accurate bioassessment. Skin burial integrity test. First, it's a visual examination alongside transcutaneous electrical resistance (TR) and transepidermal water loss (TWL) measurement needs to be accurate skin burial integrity evaluation to avoid over prediction of thermal absorption. Limitations of tritium-labeled transepidermal water flux assessment may not provide relevant absorption data for high lipophilic compounds. Potential discrepancy in burial integrity assessments for low water-soluble components. Consideration of using octanol for a more accurate skin integrity evaluation for such compounds. Overhydration due to TWF should be avoided to maintain skin surface integrity. In our lab, we standardize electrical resistance measurement to assess membrane's integrity for skin selection and qualification in our IVPT studies. Methodology briefly use an LCR mattress data bridge with stainless steel electrodes. Fixed current apply across the skin preparation. Typically, this is 100 kilohertz. Receptor and donor components filled with physiological silence 0.9% sodium chloride solution or buffer, phosphoric buffer. One electrode probe is in receptor chamber below the silent level and another in the donor chamber above the skin. Ensure not contact with the membrane. Electrical resistance can be expressed as a kilo ohm per centimeter square or kilo ohm based on the specific diffusion cells area. In our lab settings for ex vivo human skin under standardized conditions, ideal resistance should be fall within 0.8 kilo ohm and 2 kilo ohm. Lower limit used to distinguish between native and compromised skin samples. Higher resistance value, more than 0.5 kilo ohm, may be lead low or unusable flux amount. Importance of setup cutoff value because of device, frequency, current, solution's ionic strength, and skin area influence resistance measurement. Therefore, a lab-specific limit is essential due to this influence, these factors. Here is empirical IVPT data analysis. Comparison of IVPT data for 3 cream formulations on 4 different skin donors in 6 replicates each. Key findings show verifying permeation profiles based on the skin donors and the corresponding electrical resistance readings. For example, as you see, donor number 2 with resistance between 1 and 1.5 kilo ohm demonstrate the best IVPT data for expected flux profile. Donor 1 with resistance between 1.6 and the 1.8 kilo ohm showed reduced API permeation rates and extent, but still within acceptance limit. Skin section with resistance 2 and 2.55 kilo ohm exhibit unconventional flux profiles, making statistical comparison challenging. Skin donors with more than 0.5 kilo ohm showed extremely low to unmeasurable permeation profiles, rendering the data unusable. Again, the standardized value of resistance is determined in our settings, and in other labs needs to establish own acceptance range. Conducting skin donor qualification testing during the method development is recommended to ensure donor meets acceptance criteria. Using 2 independent methods like TR and the TEWA can help avoid unusable IVPT data in pilot and pivotal studies. Receptor solutions and permeation enhancers. These are 2 critical factors that must be carefully considered. The choice of the receptor fluids in vitro dermal absorption studies is critical. It must not limit permeation due to solubility issue. Ideally, its solubility should be 10 times higher than test components concentration. The receptor fluid should mimic the skin's intercellular fluid. FDA, EMA, and OECD guidance recommended aqueous, silent, and PBS buffer pH 7.4 for non-viable skin and water-soluble components, or tissue culturing medium for metabolic activity skin. Highly lipophilic substances often need additives like ionic surfactants enhance uptake. For this component, the OECD suggests solvents mixture like 50% aqueous ethanol and 5% bovine serum albumin. Non-ionic surfactants like twin 20 in PBS typically 0.1% to 1% levels have been effective. Ethanol is well-known penetration enhancing, altering the skin lipid bilayers to increase permeability. It affects hydrophilic compounds permeability, but not lipophilic ones. Pericals can alter skin permeation through the hydration or temperature changes. Occlusive and lipophilic velicles like paraffin and petrolatum reduce water loss and increase skin moisture, enhancing drug permeation. Permeation enhancer like ethanol, propylene glycol, oleic acid enhance substance penetration by temporarily reducing the skin barriers. Further, I would like to emphasize how in vitro human skin models could assess in product development and performance verification for topical lipophilic formulation. Particularly, I would like to present topical formulations with cannabinoids as a case study one. Cannabinoids are highly lipophilic molecules. Log P is 6 to 7 with very low aqueous solubilities that's susceptible to degradation, especially in solution via actions of light and temperature, as well as auto-oxidation. Formulation thus can play a critical role in enhancing permeation. The solubility and physiochemical stability of the drugs. Nevertheless, for the very lipophilic drug, which can overcome the stratum corneum barrier, the aqueous interference lower other layers will be serious obstacles to penetrate. The rate-limited steps will be, in that case, the clearance from the barrier rather than penetration of the barrier. So it is to be expected that the lipophilic drug will practically exhibit low small permeability and long lag time through the skin membranes and extensive watery dermis left in the place. The feasibility of transdermal application of cannabinoids for a management of the pain and inflammation has been demonstrated in several studies. A diverse selection of topical applied agencies has been studied in wound care, including opioids such as morphine, heroin, methadone, often combination with ketamine, capsaicin, lidocaine, and ibuprofen. Among these painkillers, morphine compounded with the hydrogels is the most researched topical agents for wound management. However, recent literature has highlighted that the significant side effects of opioids, their potentials to inhibit wound healing, and their association with fatal accidental overdosage. In response to US FDA released new guidance in February 2022 to encourage the development non-addictive alternatives for acute pain management, aim to reduce opioid exposure and mitigate addiction risk. To address this need, collaborative research was undertaken to develop transdermal cream formulations incorporated medical cannabinoids such as delta-9 THC and cannabidiol CBD to reduce opioid reliance for pain relief and wound healing. This research with particular emphasis on the early stage clinical developers for pyoderma gangrenosum, a rare inflammatory neuropathic skin diseases, often help to patients worldwide. Additionally, the development formulations could target conditions like acne, dermatitis, eczema, rheumatoid syndromes, joint pain, and muscle pain. The finite dose IVPT model was used to characterize the percutaneous absorption of cannabinoids from 4 test formulations into the through human skin. Methodology or skin absorption kinetics we are measuring using the Phonics automated diffusion system from Teledyne Hanson robotic diffusion stations. Each formulation was evaluated on 6 replicates selected from the 6 ex vivo human torso skin donors. The percutaneous absorption of cannabinoids was determined over the 48-hour period with receptor solution PBS buffer pH 7.4. Additionally, glass rod used for dosing surface wash, stratum corneum, epidermis, and dermis were recovered, assessed for drug content. The samples were analyzing for cannabinoids, THC, and CBD contents using Waters Zero TQS equity UPLC MSMS system. Analytical methods for extraction of the quantification cannabinoids employed deuterium internal standards. The following parameters were calculated: total absorption, sum of all receptor solution, rate and extent of penetration across the study period, distribution of the applied dosage in the skin, and mass balance. To ensure integrity of permeability of each skin sections, transepidermal electrical resistance TR and transepidermal water loss TWL were tested before IVPT study was conducted. Modern dermal and transdermal drug delivery technologies was used in a 4 testis formulation. Daitibas, our formulation, employs a novel nanotechnological approaches based on multi-layer lipid vesicles, which are formed by lipid bilayer and entrap both aqueous and oil phases in the form of the stabilizing emulsions. The lipophilic cannabinoids are entrapped in the oil phase and submicron emulsions and further enclosed between the phospholipid bilayers. IVPT data comparison for two. These case studies, we compare in vitro absorption profile, 3 different sponsors formulation and 1 in-house, totally 4 cream formulation. 2 most optimal carriers, Daitibas lot, and presented breakthrough transdermal, so-called, they call it liquid needle technology, showed quick and deep penetrations into and through skin. Transdermal flux and drug distribution profiles for both active CBD and THC were pretty similar for both products, and importantly, met targeted transdermal flux. This research makes a significant step toward the targeted delivery of lipophilic cannabinoids, encouraging further investigations into advanced topical delivery systems. These findings support the potentials for routine evaluation of medical cannabinoids products, contributing significant to therapeutic range determination for conditions require potent drug delivery via the skin. Further evaluate the feasibility of vitamin D3 and its derivatives for skin permeation. Vitamin D3 is a high lipophilic. Its log P is a 7.13. Here is the literature data for IVPT using ex vivo human skin and 50% ethanol. The gel and cream formulation we are prepared using chemical penetration enhancers. Penetration enhancers used was a soybean, isopropyl palmate, propylene glycol, cereal alcohol. Finding was a cream formulation. The combination of potential enhancers did not compromise skin integrity that was evaluated by TWL. Nevertheless, no significant penetration into deep skin layers or permeation through the skin was observed. While gel formulation association with a cereal alcohol and polyethylene glycol composite skin integrity compromised skin integrity within 24 hours. Vitamin D3 was detected in the stratum corneum at 4 hours and epidermis and dermis at 24 hours, but no active vitamin D3 detected in receptor fluids for both formulations. Thus, vitamin D3 did not show feasibility for transdermal use due to its high lipophilicity and lack of permeation through the skin. Therefore, further studies should be explore less lipophilic derivatives of vitamin D3 and alternatively permeation enhancers can be used. Here is a following up case study, drug calcipetrol in water insoluble lipophilic vitamin D3 analogs used topically for psoriasis and skin, the others skin and the scalp, often in combination with betamethasone. Our IVPT study objective was to evaluate the formulation and bioequivalence for NDA submissions, focus on assessing cutaneous pharmacokinetic rather than transdermal delivery. Present data shows flux profile for reference lot, already blue line, and two investigate batches, red and green lines. Permeation profiles flux, all three lots were very similar. Both maximum flux, Jmax, and total cumulative amount of active drugs permeate into the receptor solution over 48 hours are comparable. Thus, outcome is a similarity in permeation profiles between RLD and two test formulations suggests potential bioequivalence. Skin layer distribution profiles, all three lots showed very similar drug distribution across skin layers. Comparable amounts of the drugs were detected in each layers. Case study 3, this is unconventional IVPT flux profile. Some of topical products with high occlusive lipid matrix or water in oil emulsions may show continuously increasing flux over the time without a defined Jmax. As displayed on this slide, such profiles complicate statistical comparison and bioequivalence assessment. The potential solutions could be following. Dose duration studies recommended by FDA and described in USP General Chapter 70/24. Method involves exposing formulation for various duration. For example, 30 minutes, 1 hour, 2 hours, up to 8 hours. After exposure, the residue formulation is removed from the skin and the flux profile is analyzed. This approach can yield a reproducible and sensitive flux profile presented on this slide, second one. But in some very rare cases, even dose durations, such as removing products at even 30 minutes or 60 minutes after application, may not lead to the Jmax, not showing. We encountered a scenario where traditional flux profile was an initial absorption rates increases reaching Jmax and following by declining at the various time point was not observed despite collecting IVPT samples over a week. As a fact, in each consecutive time points, flux slightly and continuously increased. One of possible explanation is that the high lipophilic analyte aided by permeation enhancers quickly permeate the epidermal layers but get trapped in the hydrophilic dermis. Subsequently, a tiny amount of the analyte is released gradually over the extended period of the from the dermis, resulting as substantial increases, sustained increases in flux. In such instance, it may be advisable to consider the following recommendations. A data segmentation and piecewise analysis, segmenting data into different time intervals and analyzing these segments individually. This helps understanding the dynamics of the drug absorption into different phases. Look for time intervals where the flux stabilized to become predictable, or may use heat-separated epidermis to avoid dermis, which causes this problem. Also look for steady states, extend observation period of detect if its steady state is achieved. Aim time points providing acceptance linearity nice RSQL should be the more the 0.9, but this will need regulatory approval for sure. Next is a pilot study objectives is to confirm that the active substance permeates through the skin and validate the experimental conditions such as aperture, dosing amount, sampling time, staining rates, etc. To estimate the number of the donors and the replicates needed to power the pivotal IVPT study. Here is an example. This is an FDA approach, sample size estimation based on the pilot study result for JMAX to achieve 90% confidence interval. At least 16 skin donors and 4 replicates needed. Increasing replicates to 5 or 6 can reduce required number of the skin donors to 13 and 11 respectively. Balanced design statistical analysis is applied if samples from all donors are equal. Otherwise, an unbalanced design is recommended due to potential exclusions. EMI requirement is at least 12 donors and 2 replicates are needed for the pivotal IVPT study. Pivotal IVPT study typically topical products exhibit conventional absorption profile within the specified IVPT study duration. Normally it is like this. It is notable that EMA draft guidance specifies maximum IVPT study duration 24 hours unless membrane integrity is demonstrated for a longer period. The FDA guidance allows up to 48 hours. There is a discriminatory power requirements. FDA demonstrates the IVPT method sensitivity and selectivity. Sensitivity evaluated by modulating dose amount, dose duration, or product strength. Selectivity assessed by formulating a third part of topical products designed to be different from the reference drug. You may specify using batches with different quality attributes. Such alternation may be related to the quantitative formulations and using slightly modified process parameters. The complete omission one of or more excipients from the formulation is not supported. The negative control must be statistical different and non-equivalent to the comparator products. This is in a conclusion. IVPT is a powerful and sensitive method for quantify drug rates and percutaneous absorption. Application includes multiple areas of drug development, particularly in clinical stage to screen a selection optimal formulation. IVPT serves as a surrogate approach, facilitates development, registration, approval of topical generic products. It is useful applied to obtain biowaiver and assess bioequivalence for generic dermatological products. Efforts toward globalization, harmonization of bioequivalence standards and methodologies may continue promoting consistent regulatory requirements and facilitate cross-border approval process for generic topical products. In vitro studies are cost-effective alternatives to the cost and time-consuming clinical trials, allowing for efficient evaluation by equivalency between generic and brand name topical formulation. Thank you, Bruna. This is all today.
Thank you, Theo, for sharing your knowledge with us again. So we will now move on to the Q&A session. And I would like to remind everyone that you can submit your questions using the questions function on your screen. So we already have a few questions. First question for you, Theo. What recent advancements have been made in IVPT methodologies for assessing topical product bioequivalence?
Thank you for this question. Recent advancement in IVPT methodologies have focused on improving the accuracy, reliability, and applicability of this test for assessing bioequivalency in topical products. Could we highlight some key advancements? Development and more sophisticated skin models, including 3D skin equivalents, and use human cadavers or living skin cultures for better mimic in vivo conditions. Improved maintaining skin barrier functions in vitro, ensuring the more consistent and reliable permeation data. Second, I will call is advanced analytical techniques, utilizing more sensitive analytical methods such as high performance liquid chromatography coupled with mass spectroscopy to detect low concentration drug permeates through the skin, or use imaging technologies like microscopy or Raman spectroscopy or real-time non-invasive monitoring drug distribution in the skin. Oh, quantum dots is a very recently, it's a semiconductors, nanoparticles offering a powerful approach to studying the mechanisms of skin absorption and penetration of the topical products. Next, I will call it a standardization and validation. Development standardization protocols and guidelines for IVPT to ensure consistency across different laboratories and studies. Enhance validation process for IVPT methods focusing on reproducibility and robustness. As mentioned, updated FDA and EMA guidance recently published to support application of IVPT for development approval topical products. Use automated high throughput system, implementation automated IVPT systems to increase throughput, reduce human error, and shorten the study time. Application advanced mathematical models or adoption more sophisticated statistical models. All of these advancements helps improve credibility and utility of IVPT studies in the regulatory assessment topical bioequivalence products, I believe. Yeah.
Okay, many advancements. Next questions. What factors must be considered when selecting skin models or membrane for IVPT?
Okay, thank you for the question. Selecting the appropriate skin models is critical indeed. Obtaining reliable and meaningful IVPT data in the context of bioequivalence assessment for topical products. When selecting skin models or membranes for the in vitro permeation testing, several factors must be considered to ensure the model effectively simulates a human skin environment and provides reliable bioequivalence study. Key consideration are skin structure and composition. The selected model should closely mimic human skin barrier properties, composed and permeability characteristics. This is critical to ensure that the model maintains functional barrier integrity similar to that of human skin. Further, I could call this a source of skin, ex vivo human skin. This is often preferred. This provides the closest approximation to in vivo human conditions. Factors such as donor variability, age, anatomic size should be taken in account. Commonly used animal models include pig, rat, and mouse skin. While pig skin is considered to be more similar to human skin, differences in lipid composition and hair follicles density needs to be considered. It is important to determine a regulatory body's acceptance animal skin models for a specific BE studies, as a human skin is generally preferred. Regulatory acceptance is most critical. Different regulatory might have different reference to skin models. For example, FDA, EMA might have different guidance on acceptability of animal skin model. So it is important to refer the specific regulatory requirements for each agency. Currently, FDA and EMA do not support using animal skin for PE studies. As other factors, I could call this a practical consideration. Availability and cost has to be, data reproducibility and variability. Those factors has to be considered. Yeah.
Thank you. Next question: are there any significant regulatory differences between the FDA and the EMA regarding the IVPT data acceptance?
Oh, sure, yeah. In general, IVPT aims to establish characteristics, permission profiles of drug product test and reference, comparing relevant parameters among the assessed equivalency. Moreover, IVPT could be also just use just one study to support marketed authorization application for a new topical products. When comparing FDA and EMA guidance for a IVPT study, several important differences and similarities can be observed. These differences often reflect varied regulatory philosophies and practical consideration between two agencies when evaluated by equivalency for topical products. Let's start with a good point: harmonization efforts. Both agencies are involved in global harmonization efforts, such as ICH. Both FDA and EMA recognize IVPT studies as a valuable tool to assess bioequivalence of generic topical products, particularly in cases where the traditional clinical endpoint studies are challenging. We have guidance availability and specificity. FDA provides relatively detailed guidance on the use of IVPT within the draft guidance documented focus on specific product categories. The agency emphasized the importance and the properly validated IVPT methods, discussed and aperture study conditions and data evaluation. Proposed versions also 7024 in current USP discussed experimental designs and method development consideration for IVPT study. The EMA tends to offer more general principles regarding the in vitro study, with less detailed focus on specific IVPT of topical products. Thus, more interpretation is sometimes required when aligning study design with the EMA. Both agencies require IVPT method to be well validated and to be reproducible in relevance. This involves rigorous documentation of the method specificity, sensitivity, reproducibility, and stability indicated capability. EMA in the revised guidance specifically requested to demonstrate IVPT discriminatory test conditions using batch with a different quality attributes. The alternate lot should be statistically different to the reference lot. Guidance mentioned batch with meaningful or quantity changes made formulation or process parameters. Notably, this complete omission one or more specific experience from formulation is not supported. Other factors like the skin interpretation is quite similar for both agencies. Yeah.
Okay, thank you, Theo. I think we have time for one last question. So what are in your opinion, the critical challenges associated with IVPT studies, and how can they be mitigated?
Oh, yeah, yes, indeed, IVPT studies essential to assess the bioequivalence of topical products, but they present several critical challenges. There are key challenges and potential strategies to mitigate those. Variability skin samples. Challenges is there are inherent variability in skin samples due to differences in donor age, gender, anatomical site, health status, which can impact variability. Mitigation is a standardized skin source, carefully control, document skin's characteristics, and ensure consistent preparation and handling and storage of skin samples. Both products normally I apply on the same donors and accounted geometrical mean. Thus, until flux is measurable and presents complete absorption profiles, donor to donor variability is okay. Second, I can mention is maintaining skin integrity. Challenges is maintaining the physiological integrity of the skin samples throughout the experiment is crucial, but can be difficult, especially with prolonged testing. The resolution can be used, validated skin viability assay to ensure barrier functions before and after experiment. Employ suitable storage and handling protocols to preserve skin quality. Challenges is choosing the right skin models, human versus animal, fresh versus frozen, can be influencing study outcome and the regulatory acceptance. Application is evaluated skin models relevance to the specific products and regulatory requirements. Validate the model under study conditions to ensure it correctly correlates with the in vivo outcomes. Method standardization and validation also. Challenges is a lack of standardized procedures across the laboratories can lead inconsistent results. Mitigation is follow established guidelines and industry standards for IVPT methods development and validation. Conduct inter-laboratory studies to confirm reproducibility and reliability. Regulatory acceptance challenges also. There is, we have it. Meeting variety regulatory expectations across the different regions can be complex. Yeah, mitigation in maintain up-to-date knowledge on regulatory guidance and engage in proactively communications with regulatory bodies is important. Obtain scientific advice during the study design phase to align with the regulatory expectations. Pre-submission meetings with regulatory bodies, no, really it's very important. These factors are recommended to take into the account to overcome IVPT study challenges. Yeah.
Okay. Thank you so much, Theo. I think that concludes our webinar today.
Okay.
Big thank you to all the participants, everyone who submitted questions. As we explained before, if we didn't have time to reply to your question, we will come back to you via email. Okay. And as I mentioned earlier as well, the webinar will be available on the Teledyne Labs YouTube channel soon. So feel free to watch it on demand and share it with other people who may benefit from this content. For further information on this webinar or to access other Teledyne contact or products, please visit our website at teledynelabs.com or find us on social media. So once again, thank you very much, and we look forward to seeing you in our next webinar. Thank you.